تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي.
Welcome dear student to our channel مدرستنا 3 under the supervision of Ministry of Education. أعزائي طلاب الصف الثالث الثانوي شعبة علوم، أهلا بكم في حلقة جديدة من تدريس الأحياء باللغة الإنجليزية تحت إشراف وزارة التربية والتعليم. Okay. In this episode, inshallah, we will continue about the third line of defense that related to the last episode, first and second line of defense. In this episode, we will discuss, inshallah, third line of defense that related to acquired immunity and is divided in two types. Okay, before studying these lessons, now we are going to focus about the learning outcome that related to these lessons. Okay, that's right. Okay. By the end of this lesson, a student should be able to, number one, differentiate between the humoral immunity and cellular immunity. Number two, identify primary and secondary immune response. Finally, recognize third line defense. Okay, that's right. Okay. Follow me, and now we are going to explain the specific types of immunity. It's called acquired immunity. If the first lines and second lines failure to defense against the invasion of the microbe and pathogens, what would happen? What would happen? The third line of defense, it is highly specific or specific defense against a specific microbe and pathogen through a specific type of cells. It's called lymphocytes. What do you know about the lymphocytes? B lymphocytes, that's 3%, an average 12.5% from the total percentage of lymphocytes. T lymphocytes, that's 3%, 80% from the totals from the lymphocytes. Finally, natural killer cells, the 3% and the average 7.5% from the lymphocytes. Now we are going to define the immune response. What is the main type of immune response? Okay? Immune response is specific defense mechanism. A specific defense mechanism done by lymphocytes to resist against the microbe and pathogens, to resist and to prevent enter the microbe and the pathogens. Again, what's the main type of the immune response? A group of specific defense mechanism, okay, done by lymphocytes to prevent or resist microbes and pathogen enter your body. That's right. Okay. This acquired immunity is divided in two types. Number one, humoral immunity related to productions antibody. As mentioned previously, the B lymphocytes, it's transferred into plasma B cells and the productions antibody in lymph and transfers into blood. That means that the plasma B cells responsible for productions antibody. In this specific type of immunity, acquired immunity, specifically or especially the humoral immunity. Now we are going to divide the humoral immunity. What is the main type of humoral immunity? It is the immune response. That means that specific defense mechanism through which the B lymphocytes productions antibody to resist or prevent the microbe and pathogen enter your body. What would happen through this mechanism? Inshallah, follow me and reaching finally to understood this mechanism carefully, inshallah. Okay? Pay attention, please, and you can summarize this mechanism. The B lymphocytes, it contains receptors found in their membranes, binding with the microbe through antigens. The B lymphocytes, it contains a specific type of the protein. It's called MHC. What is the main type of MHC? Major histocompatibility complex found inside the P lymphocytes. Okay? P lymphocytes, this is the antigens found in the surface in the microbe. P lymphocytes binding with the antigen. So we found the surface of the microbe and the pathogen. Inside the B lymphocytes, MHC. The MHC transfer from inside to binding with the antigen and releasing on its surface MHC antigen complex. That means that major histocompatibility complex on the surface of P lymphocytes. At the same time, macrophages, at the same time, concurrently, the macrophages engulf the pathogens. The antigen enter to the macrophages. The macrophages contain the lysosome, secreting digestive enzymes. Dissolve the specific type of antigens and transfer into small fragments. The macrophages, as it found MHC in the B lymphocytes, found two inside the macrophages. MHC binding with the antigens, forming MHC antigen complex, transfer the surface of the macrophages. Specific types of the lymphocyte, it's called helper T cells, TH. In their membrane, there is the receptor. It's called CD4. What is meant by CD4? Cluster differentiation 4. The CD4 is binding with specific types of antigen, MHC antigen complex, in both of surface of macrophages and surface of the B lymphocytes. This specific type of MHC antigen complex activate the helper T cells through receptor CD4s and secretions a specific type of protein it's called interleukin. The interleukins activate the P lymphocytes, is divided and multiply and differentiations in two types of cells. Interleukin is secreting after binding with the MHC antigen complex in both surface of macrophages and the B lymphocytes, activates the helper T cells, secreting interleukins. Interleukins activating the B lymphocytes to divide it, multiply differentiations into a specific type of cells. Number one, plasma B cells. Number two, memory B cells. The memory B cells found in the blood approximately 20 to 30 years to recognize at the same pathogens when enter, okay, in futures. That's right. And the plasma B cells production antibody. The antibody transfer in lymph, reaching to finally in the blood, okay, binding with the antigens that found in antigens that found surface in the microbe, destroyed it completely. Okay. The humoral immunity, it's failure to defend against the virus. Why? Because the antibody Y-shaped is large molecules. So it's not penetrate the micromolecules as found in virus, so it not resist against the virus. The virus is related to specific type of acquired immunity. It's called cellular immunity. Okay, that's right. A humoral or antibody mediate immunity. This mechanism defends the body against antigens, pathogens, like bacteria, virus, and toxin present in the body fluids, blood plasma, and lymph by producing antibody. This done on the following steps. When the pathogen enter the body, carrying on its surface a specific antigen, the B lymphocytes recognize the antigen. Each B lymphocyte is very specific and can respond to single specific antigen only. When the B lymphocytes recognize its specific antigen, it's attaching itself to the antigen by using immune receptors present on surface of the P lymphocytes. That's antigen binding with proteins in the P lymphocytes and called major histocompatibility complex, MHC. That's right. At the same time, the macrophages engulf the antigen and digest it by its lysosome enzymes into fragments. These fragments binding inside the macrophages to proteins called major histocompatibility complex, MHC, Then the complex resulting from the binding between the antigen and the MHC transfer to the plasma membrane of the macrophages to be present on its surface. Okay, this is the explaining these steps. Now we are going through the drawing in figures to explain what the mechanism it occurs. Okay, that's right. Okay, follow me please in this part. Okay, second, acquired specific or adapted immunity. A, humorals or antibody mediated immunity. Previously, okay, defended the humoral immunity. It is the immune response done or through which the B lymphocytes productions antibody to prevent enter the microbe and the pathogen or fight or resist. Up to you. Okay, this is the macrophages. Membrane proteins in this macrophages. This is the antigens that represent the microbes. Lysosome found inside the macrophages. Nucleus. Inside the macrophages is specific type of the protein. It's called major histocompatibility complex. It's called MHC. The antigen, it's engulfed by the macrophages. Okay, the first stage. Second stage, the macrophages digested antigen in lysosome. Okay, as the macrophages, this is the lysosome. Transfers the antigen is small fragmented. The small fragmented binding with the MHC antigen complex and transfer in the surface of macrophages. Okay, so the antigen present macrophages display in the surface antigen fragmented on the surface receptors. That's right. Okay. Pay attention please in these figures. This is summarize the mechanism accurately. That's right. This is the macrophages. Engulf the bacteria. Antigen present in cells. Antigen fragmented. Class MHC molecules. Okay? TCRs. CD4s. CD4s that's representing the receptors cluster differentiation found in the surface of the helper cells. Binding with the MHC antigen complex. This is the MHC antigen complex. At the same time, the helper cell through receptor found in the membranes binding with the MHC antigen complexes in the surface on the B lymphocytes. In the surface on the B lymphocytes. Okay? In the surface on the B lymphocytes. This is the MHC antigen complex in both of surface of macrophages and the surface of the B lymphocytes activating the helper cells. Okay? Activating the helper cell to secretions interleukins. Okay? A specific type of interleukins activations the B lymphocytes, تمام, columns of the plasma B cells to productions antibody. Columns of the memory B cells that remain in the blood 20 to 30 years. Okay? And the antibody is transferred to the lymph or the blood to resist or prevent enter the microbe and pathogens. That's right. Okay? That's right. Cellular or mediate immunity. This is specific types of the defense mechanism. It's called cellular immunity. Cellular immunity, that means that it is the immune response done by T lymphocytes through receptors found in their membranes, okay, to give a specific response against a specific antigen. Pay attention, please, on light in this part to explaining the cellular mechanism defense. As that occurs previously in the humoral immunity, the macrophages engulf the microbe and the pathogens, okay, and transfer into small fragmented, okay. Follow me, please, in these figures, okay. This is the macrophages. On the surface of macrophages, MHC. At the same that's occur in the humoral immunity, occur in here. Through macrophages. Macrophages engulf the microbe and the pathogens. Through the MHC is transfer or display MHC antigen complex. And depending with the helper cell through receptor CD4 that found in the membrane. This is the helper cells secreting a specific type. It's called the interleukin. Interleukin activating the helper cells, secreting cytokines activate the B cells to production of the antibody. Against helper T cell secreting cytokine after activating by the helping of interleukin. Activations, the macrophages. The cytokine activation the killer T cells, that means that TC cytotoxic T cells. That's right. That means that the helper cells is activating by the affecting of the MHC antigen complex found in surface of macrophages, secreting interleukin. Interleukin activating the helper T cells. To secreting cytokine. Cytokines activation both of humoral immunity and cellular immunity. This type is related to humoral immunity. This type related to the cellular immunity. And activating the helper T cells, secreting the memory T cells. Remain in the blood as that's occurred in the memory B cells. Is formed during humoral immunity. Okay, that's right. Finally, cell-mediated immunity that related to the specific type of cells. It is called cytotoxic T cells. We studied it previously. Cytotoxic T cells is related to specific types of the implanted cells, carcinogenic cells or cancer cells, cell infected with virus. Pay attention, please, and follow me. This is the macrophages found in their surface, MHC antigen complex. Bending through receptor found in the T lymphocytes, it's called CD4s. The helper cell bending with activating macrophages. It's activating, okay? Secreting interleukin. Interleukin, in this stage, interleukin. Activating the helper T cells. Helper cells activating, secreting, this stage, cytokine. Cytokines activating the cytotoxic T cells. The cytotoxic T cells bending with the, for example, virus cells or cancer cells. When the cytotoxic T cells bending with the cancer cells or infected cells with virus, it is secreted specific to type of protein. Number one, it is called a specific type of protein. It is called perforin. Make perforating on antigen. It's destroyed completely. And the specific type of protein secreting through cytotoxic T cells, it's called lymphatic toxins. The lymphatic toxins is transferred into the nucleus. There is the specific genes destroyed it completely, the infected cells, to avoid the infected the another healthy cells. That's right. Okay, follow me again, please. Okay, inhibiting the immune response. Inhibiting the immune response after the destroy the microbe and the pathogens and eliminating the microbe and the pathogens, there is a specific type. It's called suppressor T cells, TS. TS bending is a receptor found in the membrane CD8. That means that the receptor CD8 found in both of TS and TC. TS helper T cells bending through receptor found in their membrane with both of B lymphocytes, TH, okay, and finally TC. Stop the actions of the B, productions of the antibody. Death or kill the active TH and TC, and the rest of the TC and TH storage inside the spleen and the lymph node. That's right, okay? Finally, stage of acquired primary immune response and secondary immune response. This is graph, the three in figure 21 in front of you represent the first infection and second infection. First infection represent primary immune response, okay? It's formed by helping of the B and T lymphocytes, okay? It takes about 5 to 10 days. So response, it's lower. That's comparable to the secondary immune response. Secondary immune response depending on the memory cells. In the primary immune response, okay, slow response, okay, the same symptoms, it appears as the result of the infection, but in the secondary immune, the symptoms appear destroyed completely. The memory cells, it's formed during primary response, but inactive. It's activations in the memory cells, okay? That's right. Choose the correct answer. Which of the following substance does not have a role in the action of the third line of defense? Histamine, interleukin complements perforance. Both of interleukins, complements, and perforin in the third line of defense, but histamine in second line of defense. So the best answer is A. Last questions. The injections with antibodies against the certain disease supplied the body with a long-term acquired immunity. That means that vaccines. Okay? Long-term natural immunity, a short-term artificial immunity, a short-term natural immunity. The best answer is the short-term artificial immunity. Why? Because this is the antibody related to the serum. There is a difference between the vaccines. Vaccines, it's the disease in the weak form. It's called long-term acquired immunity active, passive. Active. But that means vaccines, long-term artificials, acquired immunity, active. But the antibody serums, short-term acquired immunity, artificials, is passive. That's right. Okay. The best answer is C, short-term artificial immunity. Dear students, now are going and follow me, please, to focus about this is the mechanism both of humoral and cellular immunity through the light board. Follow me, please, and pay attention. In this episode, we will discuss or make abstract about the acquired immunity. Acquired immunity. Acquired immunity. This is the specific defense mechanism. Okay? So is the slow response. It divided two types. Okay? Number one, humoral immunity. Number two, cellular immunity. Okay, humorals, productions, antibody. Okay, related to B lymphocytes. B lymphocytes. Transfer to plasma B cells. Plasma B cells. Productions, antibody. This is a plasma B cells. Okay, plasma B cells. Plasma B cells. Productions, antibody. Memory B cells. Memory. Remains 2 to 30 years in your body. Okay? Cellulars, it is the immune response done by the T lymphocytes. T lymphocytes. What is the type of T lymphocytes? TH, receptor found in the membrane, it's called CD4. Cytotoxic T cell, TC, receptor found in their membrane, CD8. TS, sepocytic T cells, receptor found in the membrane, CD8. Okay. Finally, the primary immune response and secondary immune response, it's explaining through these graphs. Primary immune response, okay. Secondary immune response from A to B, C, okay, D, E. From A to P, production activations P and T lymphocytes. Productions memory cells and formations antibody. From B to C, this is related to sepocytic T cell, TS. Inhibit the action of P lymphocytes, T lymphocytes. Okay? This is the times between the first infections and second infection. From C to D, it is the times between the first infection and second infections. This graph again rise from D to E. It is the second line of defense by production the antibody through the memory cells, differentiation the plasma B cells to productions a large amount of antibody. So we can differentiate them by first infection, second infections. The first infections, slow response. And second infections, rapid response. Why? Because through the memory cells. It stores information about the antigens that fought by the body before. Dear student, thank it a lot for you. I will meet you again in good health. Thank it a lot for you and goodbye.
تالتة ثانوي هانت خلاص، أنا قد التحدي، خطوة كمان والبكرة هعدي. تالتة ثانوي، تالتة ثانوي.